Good morning, everyone. I am delighted to welcome you to this presentation. Today, we will be exploring the fascinating topic of the incorporation of recovery processes in semi-solid dosage forms and its impact on shelf life. I'm Dr. S.K. Singh, and I'll be guiding you through this session. We'll cover how recovery processes influence the stability and longevity of these dosage forms, which is crucial for pharmaceutical development. Let's get started and dive into this important subject. Let's start with an overview of semi-solid dosage forms, which include creams, ointments, gels, and pastes. These are complex systems that typically contain the active pharmaceutical ingredient, or API, along with various excipients, and they often have water or oil phases. Because of this complexity, they can be quite sensitive to a few key issues. For example, phase separation can occur, where the components of the formulation start to split apart, which can compromise the product's effectiveness. Microbial growth is another concern, especially since these products often contain water, making them susceptible to contamination if not properly preserved. Additionally, chemical degradation of the API or excipients can happen over time, affecting stability. It's also important to remember that recovery processes, like remixing or reformulation, can influence the product's stability and shelf life. So, understanding these factors is crucial when developing and maintaining the quality of semi-solid dosage forms, ensuring they remain safe and effective throughout their intended shelf life. Let's take a closer look at the critical quality attributes, or CQAs, specifically for semi-solids. These are key parameters we need to monitor closely to ensure product quality. First, we have the assay, which tells us the percentage of the drug content in the formulation. Then, pH is critical because it affects stability and patient comfort. Viscosity and rheology are important for the product's flow and application properties. Spreadability is another vital factor, especially for topical or semi-solid formulations, as it impacts how easily the product can be applied. Microbial limits are essential to ensure safety and prevent contamination. And finally, phase homogeneity, which ensures the product is uniform throughout. All of these attributes directly influence the stability of the product and how well it performs from the patient's perspective. So, keeping these CQAs in check is fundamental to delivering a safe, effective, and high-quality product. Now, let's discuss recovery processes. These involve methods to recover active pharmaceutical ingredients or excipients during manufacturing or reprocessing. Reprocessing means repeating the same process to correct or improve the product, while reworking involves adding an additional processing step to achieve the desired quality. Recovery also includes the recovery of APIs or excipients that might be separated or removed during production, and batch adjustments to correct any deviations. Regulatory requirements here are quite strict. Any recovery process must follow a pre-approved procedure, backed by scientific justification, demonstrating that the process does not compromise the product's quality or efficacy. An impact assessment is also necessary to understand how these recovery steps influence the final product. Overall, these procedures are vital for maintaining compliance and ensuring consistent product quality. Let's talk about some stability concerns that are specific to semi-solids. First, physical instability can be a real issue, and that includes phase separation, where different components start to separate out, and viscosity changes, which can affect how the product feels and performs. These physical changes can compromise the product's quality and consistency. Next, there's chemical instability, which involves processes like oxidation and hydrolysis. These reactions can lead to degradation of the active ingredients or the product itself, reducing effectiveness and shelf life. We also need to consider microbiological risks, especially in systems with high water activity. These environments can support microbial growth, leading to contamination and spoilage. It's important to note that recovery steps, like reprocessing or remanufacturing, can sometimes make these stability issues worse if not carefully managed. So, understanding these factors is crucial for maintaining product integrity throughout its shelf life and ensuring safety and efficacy for the end user. Now, let's take a closer look at the mechanisms of impact that recovery processes can have on the product. When we talk about reheating, for example, this can lead to accelerated degradation of certain components, which ultimately shortens the product's shelf life. Similarly, shear mixing, while useful for achieving uniformity, can cause emulsion breakdown, affecting texture and stability. Exposure to air is another critical factor. Air contact can lead to oxidation, which not only impacts quality but can also pose safety concerns. Additionally, any extra handling during recovery increases the risk of microbial contamination, which again compromises shelf life and safety. All these mechanisms, reheating, shear mixing, air exposure, and handling, are interconnected in influencing how long the product remains safe and effective to use. So, understanding these impacts is essential in developing better recovery practices to extend shelf life and maintain quality throughout the product's lifespan. Let's walk through this example calculation to understand how degradation kinetics impact shelf life. Before recovery, the degradation rate constant, K, was 0.0015 month inverse. Using that, the time for 90% degradation, T90, is calculated as 0.105 divided by 0.0015, which gives us about 70 months. Now, after recovery, the degradation rate increases to 0.004 month inverse. Repeating the calculation, 0.105 divided by 0.004 equals approximately 26.25 months. This shows that the shelf life has been reduced by roughly 60% due to reprocessing, which is quite significant. So the key takeaway here is that increased degradation rates shorten the product's shelf life considerably. Understanding these kinetics helps us better assess how processes like reprocessing can affect product stability and overall quality. Let's take a closer look at this rheology impact example. Here, we're seeing how the flow properties of a material can significantly influence its performance and processing. Rheology, as many of you know, is all about understanding how materials deform and flow under different conditions. In this example, we observe that small changes in viscosity or flow behavior can lead to notable differences in production efficiency or product quality. For instance, if a material is too viscous, it might be hard to pump or mold, while if it's too runny, it might not hold its shape properly. This highlights the importance of carefully controlling rheological properties during formulation and manufacturing. By doing so, we can optimize processes, improve product consistency, and reduce waste. Overall, this example emphasizes that understanding and managing rheology isn't just a scientific curiosity. It's a practical necessity that impacts the entire production cycle and final outcome. Now, let's talk about the microbiological impact of semisolids, such as creams and gels. These types of products are particularly susceptible to supporting microbial growth because of their semi-solid nature and moisture content. As a result, the recovery of microbes can increase over time, especially with prolonged exposure to factors like contact with equipment or storage conditions. It's important to understand that regulatory requirements play a key role here. For example, the Preservative Efficacy Test, or PET, is a critical assessment to ensure that our preservatives are effectively controlling microbial growth. Additionally, we have limits on the total viable count, which measures the number of live microorganisms present. Failure to meet these standards can lead to a reduction in the product's shelf life, potentially compromising safety and efficacy. So, maintaining microbial control isn't just about compliance. It's essential for product quality and consumer safety throughout its life cycle. Let's take a closer look at the stability study design outlined here, which follows the guidelines set by ICH. For long-term stability testing, we typically store our samples at 25 degrees Celsius with 60% relative humidity. This setup helps us understand how the product behaves under normal storage conditions over time. In contrast, The accelerated stability testing involves exposing the product to 40 degrees Celsius and 75% relative humidity. This higher temperature and humidity accelerate the degradation process, allowing us to gather data more quickly about the product's stability. Now, when we evaluate the stability, we focus on several key parameters. These include the assay, which tells us about the potency of the product, pH, to ensure it remains within acceptable limits, viscosity, especially for formulations where flow properties matter, and microbial limits, to confirm that microbial contamination remains controlled throughout the storage period. An important aspect is comparing recovered versus non-recovered batches. This helps us determine if the manufacturing process is consistent and if the product remains stable across different batches. Overall, this design ensures we have a comprehensive understanding of the product's stability profile, which is essential for maintaining quality and efficacy throughout its shelf life. Let's take a look at a hypothetical case study involving a topical antifungal cream. This example will help us understand how these products are evaluated in real-world scenarios. We'll focus on key points like the formulation, application, and effectiveness. It's important to see how these elements come together to address fungal infections effectively. By examining this case, we can connect the dots to our overall discussion on product development and patient outcomes. This kind of case study highlights the practical application of the concepts we've covered so far, reinforcing their relevance in real-life situations. Now, let's talk about the regulatory considerations we need to keep in mind during recovery. Essentially, recovery is only acceptable if there are no significant changes in the critical quality attributes, or CQAs, of the product. It's crucial that the stability profile remains consistent, meaning the product's quality and performance stay within the defined limits. Additionally, the process must be validated to ensure it consistently produces the desired quality. If any of these conditions aren't met, then we face important consequences. First, a shelf life reduction may be required, which impacts how long the product can be stored and used safely. Second, there's a risk of batch rejection, which can lead to costly delays or product loss. So the key takeaway here is that regulatory approval depends on maintaining product integrity throughout the recovery process. Deviating from these criteria could compromise quality and lead to stricter measures, so it's vital to stay aligned with these standards. In conclusion, semisolids are inherently sensitive systems, both physically and chemically, which means they require careful handling and understanding throughout their life cycle. Recovery processes, while sometimes necessary, introduce additional risks that we need to consider seriously. These risks can impact the stability and overall quality of the product, so it's crucial to support our shelf life claims with solid data. This includes stability data, which shows how the product holds up over time, kinetic modeling, to predict how the product behaves under different conditions, and microbial testing, to ensure safety and prevent contamination. Ultimately, recovery should only be deemed acceptable if we can demonstrate that there's no adverse impact on the product's stability or its shelf life. This comprehensive approach helps us maintain the integrity of semi-solid products and ensures they remain safe and effective for consumers. For further support, please contact us. We're here to help you with any questions or additional assistance you may need after this presentation. Don't hesitate to reach out. We're committed to ensuring you have all the resources necessary to succeed. Thank you so much for your time and attention today.